We're now going to get into the details of DNA replication. We're going to focus primarily on prokaryotes. For one reason, it's a little bit simpler here, but another one, this is where most of our early work on DNA replication was done. Prokaryotes, like bacteria like E. coli, are really easy to work with, so studying them and figuring out how the process worked is the first place we were able to look. So we're going to talk about how DNA replication occurs and look at all the different enzymes and proteins that are involved in this process. So I'm going to talk about this mainly in terms of there are problems that occur for DNA replication, and there's generally an enzyme that solves those problems. So that's how I like to think about it. So DNA replication is really critical because every time a cell divides, the DNA must be duplicated. We need to give identical copies of the DNA to each of those daughter cells in there. In prokaryotes and bacteria, we call this process binary fission. We talked previously about mitosis. These processes are not exactly the same, but they are very similar. In binary fission, we start with a single bacterial cell with a circular chromosome. That circular chromosome is duplicated, and then the cells divide, leading to identical daughter cells. Now, some of the differences, mitosis, we have all the machinery to pull the linear chromosomes apart and things like that. In bacteria, in most cases, the cells are directly identical. In eukaryotes, we have the whole process of development where a cell might divide and then they might differentiate or specialize into different types of cells. So some of those bits are different, but for the most part, the process is the same in terms of conceptual thought of how it's occurring. So let's look first at the problems that need to be overcome here. So first off, DNA is a double helix. We talked about that model there. That double helix, the hydrogen bonds that hold it together need to be unzipped. That is our first piece there. The DNA polymerase, that is going to need to have a free 3' OH to start replicating from. So it just can't start copying DNA. It needs something to work off of. The DNA needs to be copied accurately. It's no good making a copy if it's full of mistakes or mutations. So we'll talk about how this is achieved, this accuracy is achieved. And then we'll talk about the replication fork. This set of enzymes is all going to move in one direction, but we need two strands to get copied. So we need to replicate in two directions while moving in one direction. Might not make sense now, but I'll show you the problem in a moment. Finally, we're going to need to first fill in the gaps left by these things called RNA primers. We're going to need to fill in the RNA with DNA and then seal the gaps that are formed in there from the Okazaki fragments. Again, you won't understand what these are just yet, but they'll come into play in just a moment. Okay, so I'm going to present these problems and then show you the enzymes that solve them. Here are the enzymes that solve them. And this is a shortened list of all the things involved. We're going to focus primarily on our DNA polymerases. We'll talk about this enzyme helicase, DNA ligase, primase, and these binding proteins that are in here. So let's look at our first bit here. We're going to focus primarily on DNA polymerase 3 to begin with. So DNA polymerase, in prokaryotes, there are 3 copies of this in E. coli. Each does a slightly different job. DNA polymerase 3 is the main DNA copying enzyme. DNA polymerase, as the name implies, makes polymers of DNA. And it is the enzyme that is going to replicate DNA. It does that by using one strand of DNA as a template and building a new strand that is complementary to that. So we talked previously about complementary base pairings. That's A with T and G with C. So using the template strand, DNA polymerase is able to build a new strand of DNA that is complementary, working in a 5' to 3' direction. Okay. How does it know what to add, right? Like, how does it know that there's an A over here and a T should go into there? Well, it's not magic. It has to do with chemistry. It has to do with our hydrogen bonds that hold the bases together. So T and A can form 2 hydrogen bonds. So when you see either of those letters, DNA polymerase knows to add the complementary base there. And One of these is a single ringed molecule, the other is a double ringed. Those have to fit together properly for the DNA to be the proper size. So that's how it knows. C and G, on the other hand, form 3 hydrogen bonds. So that's how DNA polymerase knows which base to add. So that's the main enzyme. There are other enzymes that are functioning in here, but I need to talk about this whole complex here that we call the replication fork. This is the whole group of enzymes and proteins that are working together to copy a DNA strand. So on the left, we have our DNA double strand. This is our template. This is what we're starting with. We're going to build 2 new identical copies of this from this one strand. And in prokaryotes, this goes in a circle around the circular chromosome, working in 2 directions. I'm just going to focus on one of the replication forks here. They're functionally identical. Okay, so at the front of this, there's an enzyme called gyrase that helps uncoil the DNA. Don't worry too much about that. We have then coming down here, we have helicase, we have these clamp proteins, and then we have the DNA polymerase 3 that is replicating the DNA. You will see that there are 2 strands here, and one of them is called the leading strand, one of them is called the lagging strand. We'll see why that is in a moment. We have primase that makes these little RNA primers, and then we will need to stick together these little fragments that are created here. So let's get into our first problem. We need to unzip the DNA double helix. Okay, so the DNA double helix needs to be opened up. I apologize, I've flipped the diagram here. This time we're moving to the right. There's an enzyme called helicase. Helicase, its job is to unzip the hydrogen bonds that hold the DNA double helix together. To do that, it has to spend ATP to break these bonds. So it actually has to put energy in to break the bonds that are there. This unzipping process allows the DNA to open up so DNA polymerase can come in and make copies of it. Helicase, very interesting enzyme. It has this like donut shape and it slides along, unwinding the DNA as it goes along. So that is the solution to our first problem here. DNA needs to be unzipped. Well, helicase is going to do that unzipping of the DNA. Our next problem in DNA replication is the replicating itself. DNA polymerase has a little bit of a problem that it needs a free 3' OH to start replicating from. So remember we talked about how the DNA is joined together with this phosphodiester bond. It's a strong covalent bond. DNA polymerase can only add new nucleotides to this 3' OH here. And that's going to cause it to grow 5' to 3'. So if you need to copy on the other side, you need something to start off of. I'll show you what we're going to use there. But understanding this directionality, why does it matter? Well, when we want to go do the process called PCR, this directionality matters a lot to it. We got to make sure our primers are facing the right way so that our PCR will work. In this reaction, the energy is actually coming from these 2 phosphates here. They get cut off and then the base gets stuck together. So, like we said, we have our base pairings and we're going to have our anti-parallel strands. So if this is my 5' to 3' here, my 5' to 3' on the other strand runs in the opposite direction. So let's look at base pairings here and do a little practice. So DNA polymerase is going to build a new strand using the old one as the template. So let's go ahead and look at this here. Okay, so here's my strand that is a template, and where would I start building my new strand? Remember our 3 rules of DNA. In this case, we would start over here because we are anti-parallel, so this would be the 5', and we would start building in this direction. So once DNA polymerase has replicated our new strand, it runs in the anti-parallel direction, and it is complementary in sequence. Now, how does this actually start? Well, DNA polymerase can't just start doing this. So what needs to happen is some small little pieces called primers need to be added. And there is an enzyme called primase that makes short molecules of RNA that match up. What does this do? This provides the 3' end for DNA polymerase to start building off of. So this primer here is going to allow DNA polymerase to start building in a 5' to 3' direction. Now, on the other side of the strand, you will see that there are several little primers here. That's because of another problem in DNA replication. This replication fork is moving to the left, but here the DNA needs to get copied in the opposite direction, moving to the right. I will come back to that, but that is going to cause there to be what we call a leading strand, which starts from its primer and just continues replicating, and a lagging strand, which has to make multiple little primers and will result in these small fragments called Okazaki fragments. So DNA polymerase works off of the primers that are provided by the enzyme primase. So that solves our second problem here. DNA polymerase needs a free 3' OH. Primase makes those RNA primers, which give us those 3' OHs. All right, our next issue will be that DNA needs to be replicated accurately. So in bacteria, they have this long chromosome here that is circular. In the case of E. coli, it's about 4 million bases long. And the DNA is going to be replicated in two directions. The replication forks are going to move in both directions, and they will meet at the end. And we need this to be done accurately. We can't have mistakes in there. Otherwise, we would have mutations that could potentially harm the organism. This is where our enzyme, DNA polymerase 3, comes in. DNA polymerase 3 can replicate the new strand, and it's going to do so at a rate of about 1,000 bases a second. So it's going to start copying this. So DNA polymerase is replicating, and it's pretty accurate, but how accurate? If I was to say DNA polymerase 3 was 99.9% accurate, do you think that's accurate? Like, that sounds really good to us, right? Like, when I'm typing on a keyboard, I am nowhere near 99.9% accurate. But if you think about the math here, 99.9% accuracy means that every 1,000 bases, there is an error. When you look at a bacterial genome with about 4 million bases, that is a whole lot of errors that are in there. That's an unacceptable number of errors. So how do we replicate the DNA accurately? Well, polymerase 3 is pretty good, but we have to have some other systems to help with this. Those include DNA polymerase 1, which is an enzyme that can proofread the DNA. We'll also see that it plays an important role in filling in the Okazaki fragments in a moment. Additionally, there are DNA repair enzymes that come in and look for mutations that have occurred and fix those problems that have occurred. All right, so we've copied the DNA quickly, 1,000 bases a second. That's pretty fast. DNA polymerase 3, pretty good at copying accurately, but we also have DNA polymerase 1 that comes in and proofreads, and there are DNA repair enzymes that are going to fix damage that's in there. I will talk more about these later in the mutation section. We have the replication fork next, moving in one direction, but synthesis of the DNA, making new copies, needs to happen in two directions. So what do I mean by this? All right, let's look at this diagram here. We're looking at the replication fork, and it is moving to the left, right? Here is our DNA helicase, unzipping the DNA. DNA polymerase 1 here is moving 5' to 3', right to left. That's what we call the leading strand. It just moves in one direction. On the other side, though, we need to replicate from left to right. But I told you, everything's moving to the left. How do we deal with that? Well, the DNA is going to get replicated in small little pieces. And these pieces are going to basically be little chunks. We call these Okazaki fragments. How the DNA deals with this? Well, we can take the DNA and loop it around, and we get the DNA in the right orientation. So if we take our DNA here and we take the 5' to 3' is going that way, 5' is right there, and 3' is there. If I take this DNA and I loop it around, it's now going 5' to 3' in the correct direction. Excuse my terrible drawing here, but it's the best I can do with my mouse. So we're going to make a loop, and now we can copy in the correct direction. So let's take a look at an animation that'll show this much better than my terrible diagram.
Before a cell can divide in two, it must first duplicate all of its DNA so that it can pass an exact copy to each daughter cell. DNA replication is carried out by a molecular machine called the replisome that pulls apart the double helix and makes an exact copy of each strand. The DNA to be copied enters the production line from the left. The blue ring-shaped molecule is the enzyme helicase, which unwinds the double helix into two strands. The leading strand is copied continuously. The lagging strand runs in the opposite direction with a more complicated copying mechanism. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So hopefully you noticed that the lagging strand, which was on the top of that diagram, formed those little loops. Every time it formed one of those loops, a new primer needed to be added, and DNA polymerase III was added to replicate. Then when it ran into the last little chunk, then it finished and a new loop was made. Primase makes the primer, and then DNA polymerase III comes in. These fragments are called Okazaki fragments, and you'll notice there's a little bit of a problem here because, one, there's a bunch of RNA in our DNA. We've already said RNA is a little less stable than DNA, so we need to fix that issue. And also there's some gaps here in the DNA backbone. That's bad news because it means the DNA is not fully intact. So we've replicated our DNA, but in the process, it has created these pieces called Okazaki fragments. So our leading strand was fine. There was no issue there, but the lagging strand creates those Okazaki fragments. So we need to fix two issues that are created by that. We need to fill in the RNA primers and then seal the gaps from the Okazaki fragments. Okay, this is where DNA polymerase I, which we've talked about previously, plays another role. So DNA polymerase I has what we call exonuclease activity. It can actually chew up bases and replace them. It does this when it proofreads, but it also does this when it takes out the RNA primers. So here's our RNA primer in green. DNA polymerase I comes in and removes that RNA and replaces it with DNA. While it's doing that, it does its proofreading job, right? So here are Okazaki fragments, and here we've replaced the primer with DNA. So DNA polymerase I comes in and replaces those RNA bits with DNA. That problem is solved. We still have a gap, though, in the backbone here. So we still need to fill in the gaps in the DNA backbone that have been left behind. All right, so there's all these little gaps in here, and if we didn't fill them in, the DNA could break. It could snap in half. There's an enzyme called DNA ligase, which comes in and fixes the phosphodiester bond. It fills those gaps in. So why do I talk about DNA ligase? Why am I talking about any of this? Well, number one, I think it's super cool to understand how your DNA is replicated. But what I think is more important is let's think about how we can use this. Okay, in this replication fork, I've told you about a couple different enzymes. I've told you about DNA polymerase. That is an enzyme that can make copies of DNA. Well, if I'm a scientist and I want to make copies of DNA in a test tube for my research or for testing for a disease or something like this, I can steal DNA polymerase and put it in a test tube, and now I can copy DNA in a test tube. That's exactly what scientists have done in the process called PCR, polymerase chain reaction. We will learn about that. We will do that in this course. How about DNA ligase? Why talk about DNA ligase? Well, DNA ligase has this cool ability to basically glue pieces of DNA together. Now, if you think back, I told you an example of how transformation, that process that was discovered by Griffith, was important to humans. Remember, I said we could take bacterial DNA, that little plasmid, and we could take human DNA, like the gene for insulin, and put that into the plasmid. Well, how do you think we paste the DNA together? That's right, we use DNA ligase, okay? DNA ligase is like our sticky tape or our glue that we can glue bits of DNA together. So we can make what we call recombinant DNA. We can take DNA from different sources and stick it together using DNA ligase. This is a common theme in biology. Scientists are clever, but you know what's clever? Millions and billions of years of evolution. So most of the problems that we have as scientists can be solved by things that we find out there in the natural world. That's why it's so important to study these obscure processes, right? You might be going, why do I care about DNA replication? Because it enables us to do all these different things with biological organisms. So understanding this process is really key to understanding biotechnology. All right, so we've solved our problems here. DNA ligase comes in, fills in the gaps in the phosphodiester backbone, and we're pretty good. So in bacterial replication, there's a few other key events that are in there. It starts at a specific sequence. We call that the origin of replication. What that sequence is varies by organism, but there is a start, and it goes around, and then it stops. We saw that DNA polymerase was adding our new bases there. We saw RNA primers were needed for it to get started. Polymerase builds 5' to 3', and the problem was the replication fork only moved in one direction. So we saw leading and lagging strands came from that. Once replication was completed, we got to get rid of those RNA primers. We replaced them with DNA. Polymerase 1 did that, and then we had those gaps in the phosphodiester backbone. DNA ligase comes in and helps fill in those gaps. And like I said, that's real useful if you're a biologist and you want to cut and paste DNA. We'll talk later about the enzymes used to cut DNA. Of course, we're going to steal those from nature as well. So every enzyme and protein here solved the problem. The single-stranded binding proteins, those play a small role. They just hold the DNA open once helicase opens it up. So don't worry about them too much, but you should know how each of these enzymes here functions in the replication process. Now, some of you are going to wonder, what about DNA polymerase II? That's a great question. We're not fully sure everything it does. It's really hard to study these things because the way that scientists usually study how processes work in an organism is they break them. If you break DNA replication, you really don't understand how it's working because your organism does not reproduce, right? So it's really difficult. As far as we can tell, right now, current evidence suggests that DNA polymerase II is involved in DNA repair at some level. It might also be, and this is a second hypothesis, that DNA polymerase II is like a backup copy. If there were to be a mutation in polymerase 1, DNA polymerase II might be able to fill in and cover for DNA polymerase 1 if that enzyme got broken somehow. So I know someone's going to ask about that. They always do. All right, so let's review. The enzyme that synthesizes, that means makes, a new strand of DNA nucleotides from an existing DNA strand is called, go ahead and pause the video, think about this one. All right, our enzyme that makes new DNA from an existing DNA strand is called DNA polymerase. DNA makes polymers of DNA. Does what the name says. Okay, so DNA replication. This is going to happen every time the cell divides. And we thought of it as a series of problems that are solved by different enzymes. DNA needs to be unzipped. Helicase unzips the DNA. DNA polymerase needs a free 3' OH to start replicating from. Primase added RNA primers in there for our DNA polymerase to start copying from. DNA needs to be replicated accurately. DNA polymerase III does most of this. It makes the new polymers, but it makes some mistakes. So DNA polymerase I comes and proofreads, and DNA repair enzymes also come in and help fix any mistakes that are in there. We saw that the replication fork moves in one direction, but we got to make DNA in both directions. So we saw that these looping strands on the lagging side got created, but that made little Okazaki fragments, which needed to be fixed, basically. So we need to fill in the RNA primers. DNA polymerase I came in and cuts them out and fills them in with DNA. And then we need to seal the gaps from the Okazaki fragments. That's where DNA ligase comes in, and it fills in those gaps in the phosphodiester backbone. All right, that's how DNA is replicated. Super useful because when we go to do DNA replication in a test tube, we're going to need to understand those processes there. All right, in the next section, we will talk about how eukaryotes replicate their DNA. There's a few differences there.